Welcome to Tech Tuesday. All of you joining us today, both online and here at Go to 10. This is Tech Tuesday with Sectra, and Tech Tuesday is a collaboration between Linköping Science Park, Combitech, Ericsson, SICK, Nira Dynamics, and Sectra. The event is held around every third Tuesday. My name is Frida Enebro, and I work as Communications and Events Officer at Linköping Science Park. If you have any questions after this seminar, feel free to ask them afterwards. And those of you online, just write your questions in the chat and we will ask them for you. This presentation will be recorded and put on our website later on. Today we will hear Fredrik Lysholm, General Manager, Genomics IT, talk about clinical genomics, a completely new field of diagnostics. Welcome.
Okay, thank you very much. Yeah, I'm gonna talk about a slightly complex subject called genomics, precision medicine, and get into a little bit what Sectra is doing within this space. So we're in a diagnostic field, and as of such, as I think most people these days wanted to find out what does AI think about this. So I prompted DALL-E with cancer genomics and got an image. Well, a few of them. I picked this one. It's kind of cool. Looks like something out of the Spider-Man movie, perhaps with some DNA sneaking in there, giving us superpowers. Well, that is not what we're doing with genomics. Would have been cool, but it's not. So my name is Fredrik Lysholm, and I'm going to hopefully guide you a little bit of what we're doing within this space. But first of all, I think it's nice to talk a little bit about Sectra. I'm not sure how known we are in Mjärdevi. I think quite known. We're the company building over there when you come in from Lambohov. So what does Sectra do then? Well, very brief. I mean, within the company, we have two main business lines, one being the medical side, which I'm working at, and the other one being secure transmissions, or the IT security part. And I think you heard Leif Nixon at Tech Tuesday before talking a little bit about what we do in that space. Alongside of these two main business lines, we also have what we call business innovation. It's within that space we're trying out a few new things, giving birth to our upcoming areas. And within that space, that's where we find this new diagnostic area, which we call Genomics IT. So with that in mind, I wanted just to also talk about what we do at the company. So sort of our company philosophy. We want to target hard problems that society must solve. And in addressing those, we think we want to do that with happy customers. And in order to do that, we believe that we must have happy employees. And if you have those two, and you bet on your employees, and you do so with some grit to get there, and also with some reasonable cost control, we think that we will generate value in long term, and actually then shareholders will be happy. We have still at the company our founders owning the company, so it's a company which I dare to say is quite resilient towards outside pressure of the market, trying to get us to be more profitable and so forth. We're sort of staying on our course and betting on happy customers and happy employees. So what are hard problems then to solve? I think you all can come up with quite a few of them. So there are many hard problems. But one of those are actually that we're getting older, and that's what I'm getting at with this graph. This is the population of Sweden, and it doesn't look that bad. Perhaps it's a quite thin population pyramid, Going back 50 years, it looked better, and we can actually see that in sort of the growth graph here of the population of Sweden. So we're getting older, and this is sort of not as steep population increase anymore. So we have to cope with that. That means less people that can support the growing older population. Well, actually, it turns out that Sweden and Scandinavian countries are really, really good at this. We're having reasonable childcare policies, and we actually are able to sustain this quite good. If we compare to a company like Germany, this is the economic engine of Europe, or often referred to as such. They're facing population collapse instead. This is going to pose a huge problem. And actually, this bracket, these guys in the facing retirement age, they're gonna get retired, and then you'll have these guys having to support them with a decreasing population. That's gonna be hard. That's gonna put a lot of strain on healthcare. And this is not like that Germany is very, very special. Actually, Italy looks even worse. China is sort of, it's outside of Europe. They're sort of at the same pace. They're also facing population collapse according to the official statistics. If you look at some people talking about the unofficial statistics, they claim that China is already decreasing. I don't know. The other problem is that as we get older, we're starting to cost more. We need more healthcare. And healthcare is not cheap. It's not even getting cheaper. We're not good at this. I mean, I'm going to get to that, but our mission is to help healthcare become more efficient, to cost less and provide the same. Well, actually, we're providing more and more all the time, and that's one of the drivers driving cost. So healthcare is costing more and more. So this is an equation that doesn't add up, and that sort of connects to our mission statement, where we want to increase effectiveness of healthcare while maintaining and increasing quality of the patient care. So, why I'm talking about this? Well, we're actually, or rather, how does this connect to genomics and precision medicine? Well, actually, precision medicine is adding complexity to healthcare. It's making this equation even harder. We're employing a lot of fancy techniques to get at this. So what's precision medicine then? Well, if we put that in a cancer case that this is illustrating alongside traditional medicine is about treating a cancer as a cancer. Well, it's a cancer. We can do radiation, we can do surgery, and we can do chemotherapy. That's treating it all the same. It doesn't really matter what type of cancer it is, or how the patient can handle this. And that is effective, but limited so, and effective for some cancers, but far from all. That has given birth to what's called, sometimes referred to personalized medicine or precision medicine, where we can employ new techniques that are narrow to really target the underlying cause of cancer. And why have I circled genetics here? So these techniques include immunotherapy or different types of targeted therapies. But in order to know which patient to treat, we have to take this new realm of knowledge into account. So we've long known that a lot of patients react different to the same treatment. We have the same treatment to patients that look the same, they have the same disease, but they react totally different to that. It's been known for ages. We haven't known why. We've had a lot of theories, of course, and that have been growing throughout the 20th century, of course, that knowledge, and we're gaining more and more. I think at the turn of the century, we determined the human genome. It was a big project. It cost $3 billion, so really expensive. that sort of gave birth to this new era of information where we getting molecular information. Since then, cost has fallen dramatically, and we're down today with, as it says in the bottom here, around roughly $200 or $100 to sequence a human genome. That, compared to actually costs, there are costs associated with treating patients all the same. They had adverse effects, and we have to help them with those effects. So that actually means that it's not only better for the patients, it makes economic sense to start employ these techniques to help patients. And this is happening, and as everything these days is happening really, really fast. So I don't want you to sort of read into the graphs. It's a number of FDA-approved narrow therapy drugs. It's the amount of money going into precision medicine. But you see all the trends. It's sort of hockey sticking. It's a lot that happens in this area. So that a little bit of background of precision medicine. So how does genomics connect to all this? Well, in order to say that, we have to understand what genomics is. So I'll give you sort of a very brief introduction to this. This is an area which we can deep dive, and we can have probably a 2-hour session on just the intricacies of the cell and what happens. I think most people these days know about DNA. You've heard the concept, you understand that. It's often referred to as the blueprint of the cell. It's what's in the nucleus, and the cell utilizes this information in order to produce the micromachineries of the cell, the proteins that actually do stuff. That's a very simplified model of the cell, and actually DNA is much more complicated than that, has a structure. And we see that structure sometimes. It's a chromosome. That's when the cell splits. We can see that structure, and we can actually extract knowledge from that. But the chromosome, in turn, is a complex structure of folded nucleosomes, which are DNA twisted around histones that pack the DNA into this structure. And to give you an idea of how small this is, we see the cell that's about 30 to 100 microns. We can see that in a microscope. The chromosome is 5 to 10 microns. We can actually see that as well in a microscope, and that's what we've done for long. But going into this kind of structures, we're far below sort of the threshold here is 500 nanometers, half a micron. So we can't see this anymore. So we have to employ other techniques to determine what's this sequence, what's the DNA sequence, and what does that mean for the cell. So why do we need this, or what's important? Well, actually, there's a concept called liquid biopsy. Perhaps some of you have heard of the company Theranos and Elizabeth Holmes. I see some recognizing smiles here in the audience. Well, she had this idea, or sold this idea, that with a single drop of blood, we can answer all the questions that we need. She was in Silicon Valley, and actually, this is all theoretically possible, and that's why I think it worked to sell the idea. Now I think she's got 11 years for fraud or something like that, and is facing a jail sentence because it didn't pan out. So why is that? Well, this isn't really possible yet. Instruments has to be gain a little bit more sensitivity, and actually there are complex things. I mean, we can detect a lot in the blood. Actually, most pathology, or I think all pathology, will leak into the blood, and we will be able to pick that up. But it doesn't mean that it is pathology. Finding cancer DNA, sort of, I have to put that in quotes because it's gross, like we can't really say that, but we can find mutations that look like cancer in the DNA. It might just mean that your immune response is working. It's killed that cell off and it would leak out into the blood. So it doesn't mean that you have cancer, and we have to figure out how we can sort of balance that in terms of what we're seeing. But this is sort of a future, and we will eventually be able to detect everything from blood. So that's how we can use genomics. What about Sectra then? What are we doing? In the medical side, I said we're a company of diagnostic IT, or perhaps I didn't say that, but we are. And we've been working with imaging diagnostics for all time, and that's what we're doing today, where sort of genomics is not images anymore, because I told you it's below sort of the threshold of what we can have in the microscope. So we're in this middle space, but precision medicine told us that actually we need this information in order to ascribe these narrow therapies that will save the patient's life. So we want to go further. So this is what we have today. I mean, we have a lot of imaging techniques. We can do everything from ultrasound. So this is a breast case where we have an ultrasound. We detected something that looks a little bit strange here. We did mammography. This mass here is very suspicious, and then we cut that out. This is the mastectomy and radiographic image of that. This is the whole slide, and then this is a tumor slide. We can have that alongside and to start to sort of build that story within the system and connect the information, but we're lacking molecular. So really what we want to do is to be able to provide the whole plethora of diagnostic information in order to help within cancer. So what's clinical diagnostics like then? I think, well, this sort of describes it a little bit. If you zoom out, it's about producing data and interpret that data so we get actionable information from that data. And we've had that journey. We started 30 years ago with radiology, helping them to digitalize and working with them and sort of building a very efficient workflow that ensures that we can help the patients and help them quickly. Because if we can't help patients quickly, I mean, they suffer from not getting an answer, of course, but we're also building a queue. So clinical diagnostics is a lot about, think about it as a factory. Radiology department is much like a factory. If we can't keep patients flowing through that machine, it's gonna start to build up, and then patients are not getting their diagnostics and not getting their care. So it's a lot about efficiency, and that's what we're working with Sectra to help the hospital institutions to build good workflows. And workflows, as I said, with precision medicine is getting more complex. And when we went into digitalizing pathology, the sort of discipline where you have cells that you look at on a slide that I showed you before, we realized that they are facing the same problems as radiology did. There's a lot of like similarities, even though they never talk to each other. So we're like, hey, come look at what they're doing in the radiology department. You'll learn something. And the story with genomics is sort of the same. They are digital from the beginning, but they're coming from academia, and they need to bring that into a clinical setting and do mass production of diagnostic information. So that's sort of how that ties in with our products and how we're helping to build a coherent solution. So a little bit about what genomics really is then. Well, we have this DNA, right? What we really do is we take that biopsy that we had into the pathology department, a portion of that we need to take out, extract DNA from it, which means adding chemical processes to get the information out of the cell nucleus. That is fragmented and put on a big instrument. It's called a sequencer. And I won't go into the details of what that one does. It's different kinds, and you can find it on YouTube if you want to. But you do sequence that data, which means you determine the sort of sequence of the A, T, and C, and G. That is information, or data, but it's not something that we can work with. So we want to take these unaligned sequences, that's just fragments, and we want to align those back to a reference genome. And when we talk about bioinformatics and so forth, these are referred to as reads. So a reference genome is then sort of what a normal patient looks like. And to just give you some idea of how much information that is, I think this slide is kind of cool. So if we print something at size 12, and I think most IT people understand what the 12 pixels is, it's not a lot, but still it stretches on for between Boston and Houston, so it's quite far. So aligning that back to the reference genome, so we have the lab part, we do the sequencer, and we provide a computational job here to map those to the reference genome. Now we're starting to get information out of this. All these reads cover some part, and we can actually start seeing how is that different. So this alignment is stored in a format called BAM, typically, which is short for binary alignment map. So this is the huge creativity of the bioinformatics people, which I'm one of. Then you can start extract what's different, and then you get into a set of variants, which is stored in a VCF file, which is just a variant call format. So again, huge creativity. That, alongside with a lot of other information that you need, we need to understand, which research for years has helped us with. What does this portion of the DNA do? Is that a gene coding for a protein? And how might this change affect the protein, affect the cell? So we have a lot of data sources. We have the reference genome. We have the background population of different type of variants. We have knowledge about which variants are seen in cancer and seen in different types of cancers. That put together takes what we typically get is about 3 million variants for a sequencing, so quite a lot. But all this information together puts that into a short list, which a human can start to look at and interpret and find what's really important. So it takes those 3 million down to perhaps 100. Out of those 100, we, or a variant interpreter, will look at that and find the short list of what's really important. And that goes into a molecular report, which will help the oncologist to go further. So typically we get a few variants which are known to be pathogenic or likely pathogenic. These in turn will suggest the different types of treatments that we can give that patient to help them. So that is the Sectra initiative we're doing. We're currently working, and Sectra is doing this all the time. That's how we do stuff. We're working with our customers to solve their needs. So currently we're working with Penn Medicine. It's an Ivy League college medical center in the US, and one of the most prominent cancer centers in the world. And their chief pathologist, Mike Feldman, he heard our CEO talking about our vision, how we are thinking about integrated diagnostics, how all these pieces fit together, and thought that that really aligned with his ideas of what they needed. So we formed this co-development agreement, and now we're working together to extend the Sectra solution to cover this new space. And that's why I'm here today, to talk about this new diagnostic unit that we formed at Sectra. I thought I'd round off with giving a little bit of the technical perspective. Hopefully some of you are interested in those aspects as well. Well, they're producing a lot of data. It's about 2 gigabytes per case, and I often get that question, how much data is in genomics? A lot of people heard it. It's a lot of data. Well, actually, it can be more, it can be less. I usually respond with, well, how much data do you want to have? Because it turns out that 1 gram of tissue, it's about 1 billion cells, and all these cells in a cancer are different. And it's much more information than we can look at. So we look at a fraction of that. So sequencing more gives us more data, which gives us more sort of statistical knowledge about what's the underlying drivers. And going deeper, which we call getting more reads, it's getting more information. So Penn, currently they're sequencing so that each position in the genome, in the panel that they're looking at, and they're looking at a part of the genome, that one is covered by at least 500 reads. So 500 small fragments covering each position. So it's a lot of data. And their sort of back catalog of data, it's in the range of petabytes. Sectra is providing this, or want to provide this as a cloud service, a multi-tenant solution. We're building this with a zero footprint, which means web technology, viewer using TypeScript React as a front end and .NET Core as a back end. And this is a service offer from Sectra, and me and my team were evolving into a DevOps organization. So that's a little bit of what we have in front of us. And when I think about the future, so again, I asked Dolly to give me something, give me an artistic painting of a road that leads into an unknown future, and this is what I got, which I think is pretty neat. I think it's safe to say that it is an unknown future, and that this is the fastest, by far, moving field within clinical diagnostics as of today. It's happening so much. We're getting new instruments, we're getting new techniques. There's a lot of cool stuff around the corner where we can look at very narrow parts of a sample and get exactly what that's comprised of, and we can compare that to different other parts, and there's a lot of things happening. Whether it's going to hit clinical, like mass production, as I talked about earlier, I don't know. But from Sectra side, we will continue to work in close collaboration with our customers, because that's what we've done. That's how we try to stay on top of things and follow sort of where modern clinical diagnostics takes us. But as at Sectra, I think we always remember it's in the end it's about the patient, to provide faster and better diagnostics, and that in turn leads to personalized and better patient care. And with our systems, with our platform that can provide all this within the same system, we think that we can build feedback loops to help improve healthcare, because that's also a problem that we're facing today with all the complexity. How do we learn from all this data? And to provide this at a lower cost and sort of try to at least seemingly decrease this complexity. Because in the end, I mean, we'll all end up there. We'll all become a patient or someone we love or know. Right. And that's all for me today. Thank you.
Does anyone have any questions?
Yeah.
So it's very great to be here. And when you said it's 2 GB per case, is it a whole genome sequencing data that you will be getting, or a targeted genomic sequencing?
So mostly in cancer today, if there's not a research perspective as well, they will do a gene panel. So one picks out somewhere around 300 to 500 genes that we know are involved in cancer and actually is treatable.
Okay.
And then you amplify those specific parts. So that's what you typically do. The next level of that is do whole exome sequencing, which means that we take all the parts that we know are connected to our genes and connected to protein synthesis. And then you can do whole genome sequencing. But mostly it's panel sequencing or whole exome sequencing in clinical genomics in cancer today.
And in terms of service, so you will be analyzing this data, or will you be like segregating it into a certain structure and then presenting it back to them?
So what our solution exactly will do is to handle the BAM and the VCF file to be able to produce this shortlist that the clinical interpreter would like to look at. So we're relying on, this is developing all over the place, different bioinformatic pipelines that do a lot of the computational job at arriving on what's the relevant sort of short set that we want to look at. At this current point, Sectra is not considering providing that bioinformatic pipeline.
Okay, thank you.
Thank you for the cool works you're doing there. I have a question regarding what happens after diagnosis. So maybe it's not exactly your field, but I'm very curious. So, okay, we found out these genes are bad and like we have problem with this patient. Then what's the next step in the precision medicine? How do you treat that bad genes? Like how do you, after you find out the problem, how do you deal with it?
Right, and then we have to distinguish between what's called a germline mutation and a somatic mutation. Germline meaning what you got from your parents. So that's something that's fairly untreatable today. I mean, we can provide drugs that sort of work around some of the germline effects that you have. When we look at them in cancer, it's called somatic mutation, meaning acquired mutation. That's the mutation that the cancer have, sort of understanding the weaknesses of that cancer. Then it's about directing treatment with that knowledge, i.e. providing therapeutics that target these pathways that's sort of leading to cancer, meaning killing off those exact cells. So it's therapy in that sense. It's not about editing the genome, but rather sort of getting to know the cancer weaknesses and killing those cells, helping the body to kill those cells.
This happens like with drugs, right? You just give that patient some drugs which are specifically targeting those cells.
Yes.
Okay.
And hopefully the patient will get better, or otherwise you have to monitor and come back and do rediagnostics and find other means to target that cancer. And we know with cancer and mutations in general, and I think we all as a society have seen this with COVID, that if you provide external pressure on an evolutionary process, it will shift. And that's what happened often in cancer as well. Unfortunately, you have a very diverse tumor with a lot of different mutation, and you start treating some of them, meaning selecting for the other ones. And then it's sort of a whack-a-mole game. So it's tough.
Another question. Thank you.
Hi. I was particularly interested in the whole tech stack that we just glanced a little bit at. I have a background in distributed and pervasive system. So my curiosity is more like why specifically these technologies? Is it because it's a technology you're more familiar with, or because it has some special secret sauce that will help with the final product that you're trying to achieve?
Right, it's a good question. I'd say that Sectra as a company is quite .NET centric. We've been a Microsoft partner for quite a while. I wouldn't say that we picked .NET because it has features that we couldn't find elsewhere. Although, I mean, I personally think it's a good platform to build on. It's very competent and easy to work with. And nowadays with .NET Core, you can run it on a Linux machine as well. So it's kind of cool. In terms of building something in front end, well, same story again. I don't think that React does something that other platforms can't do. But it's a fairly good platform that we're used to, so that's why. And visualizing a lot of data in that sense means that we actually utilize sort of the React framework in that sense with virtual DOM updates. So that's one of the features that we actually need. Okay, did that answer your question? Or? Yeah, of course. Well, yeah, I know. It's about. It's a little bit like religion. Like, what do you choose? I mean, and.
No, it was like my main question was if you believe it's the tool that is perfect, like not perfect, but good enough for the job you're doing, and your answer is yes.
Yeah, that's correct. I think so. And I also think, I mean, being in the cloud is also something we didn't perhaps explicitly ask for, but that's also something that we believe strongly in because it's a lot of data that we need to store. I didn't get into the details, but also being able to provide sort of the computational backend that the cloud provides to be able to run these jobs, which are long-running jobs on very specific hardware, which is cumbersome for the hospital IT to provide locally, which is also something that we aim to be able to provide to our customers.
You also may probably concern on be sure that the data is stored in the proper spot. So you don't want to have one organization data leaking into another. And
yes, of course. Yeah. It's sensitive data for sure. It is.
Do you have another question? Okay. Because I thought this was a really interesting discussion, and I'm so sorry I took the microphone from you.
Hi, I have a question. You talked about liquid biopsies. Do you think that in the future that the pathology step will be excluded so that you don't need any invasive samples, that you can connect with the genomics and the radiology, so you can look at images from, for example, the brain or the liver or whatever, and then just take a liquid biopsy, blood, cerebrospinal fluid or plasma or something like that, and then run it in an NGS sequencing and then treat it without the biopsies and the scanning of the pathology samples?
It's a sensitive question. It's a question that you sort of avoid when you talk to your customers because they are pathologists and radiologists. But sure, I mean, somewhere along those lines, I think it might sort of cannibalize on pathology as a diagnostic discipline, meaning reducing the volumes that will actually end up underneath a microscope back in the days, but a scanner these days that digitalize that image. Perhaps it won't completely rid ourselves of that need. I mean, we still are helped by radiology. We might have sort of a liquid biopsy screening in the far future, where we will find out, well, I'm sorry to say that you probably have cancer. You should go and see a radiology department to find out where it is, because it will tell us a lot of information of that. We will actually see what type of cell cancer type it is, probably where it originated from. So we will have a good idea of where it sits, but we still want to figure out if it's going to be possible to operate on the patient, and do we want to, or can we provide radiotherapy of sorts? So radiology will certainly be there. Pathology has a case in when we do surgery on a patient. We want to sort of follow, like if you do breast surgery, for instance, you follow sentinel nodes. So you do that, and then you do freeze sections, and you put those at a microscope, and then you continue along the way if you still find pathology, and to take additional lymph nodes. So it's a medical discipline that will prevail, but perhaps in the future it might be less important. But it's speculation, of course.
Thank you so much, Fredrik.
Thank you.
So this was the last Tech Tuesday of this season, but we will be back again in the fall, and we hope you will join us then. If you have any feedback, we'd love to hear from you. Just email me back. You all got an email from me earlier today, so just write back. Thank you so much for coming today. See you in the fall.